previous years. My name is Yoram. Maybe okay, you'll have my name on the presentation in a moment. Before we start, I want to get some feeling for the background of people here. So, how many people here studied in their undergraduate topics which involves math, physics, math, computer science? Okay, that's about half the class. And how many didn't? Okay. No, I just really wanted to check that it sums up to everyone. Okay, this is the usual situation in this course, and what this means is that the class is very homogeneous in this respect. And this is, I suppose that all the people, or at least most people here, also took Neural Networks 1. And so you already know that for those of you who don't have a strong math background, these are difficult courses. They are designed in a way that you should be able to learn them, but it's hard work. Okay, so it's really important to do all the exercises, to spend enough time going through the concepts, both in math and the concepts that go beyond the math, and that's part of the difficulty. And so what we what we try to do to help, I hope that my presentation will go up. Okay, what we try to do in order to help to some extent is that in addition to the formal hours of the course, which are Tuesday from 12 to 2, Thursday from 12 to 1, and the TA session, which is from 1 to 4, Neta, the teaching assistant, will have an extra hour, and this will be after the regular TA session. And the goal of this hour is to go through material based on your requests, okay? But to go through it slowly, sometimes to the mathematical aspects, really based on what you feel is needed. And so Neta will explain to you more how she plans to do this. I'm not sure that she's going to do it this week. In any case, she'll discuss it with you. Okay, I am in the university. I'm part of the physics department of the Rakach Institute and ELSEG. And so physically I sit in both places. And this semester I'll be at the Rakach Institute in the Danziger B building from Sunday to Monday, and I plan to be here in this building, on this floor, from Tuesday to Thursday. So you're very welcome to stop by and talk with me. I'm not going to have a formal hour but just feel free to stop by. In the worst case, I'll tell you that we should schedule some time later. Okay, so you have my contact information here. Hours of the course. We will have assignments. Assignments are very important. They're going to be 50% of the grade. And the Moodle site is still not up, but I will put it on later today. Now, one thing I want to discuss, not formal thing again, is extra hours. So we lost 1 week. In principle, we don't necessarily, according to the instructions of the management of the university, we're not supposed to complement them, but I think I would like to complement them at least a little bit, at least in part, and also we will miss some classes later on due to ELSEC events, so we will need to set up a time in order to have complementary hours. Probably something like it will happen 4 times during the whole semester. And so it would be helpful if we can just check now. I think the best options when we want to do this is to just put it next to the class. So is this okay with everyone? Tuesday 11 to 12? Okay or not okay?
Okay, I think either Tuesday early.
Okay, let's do it this time? Anyone object? Okay. This time? Thursday 11 to 12? Thursday 2 to 3?
I think it is.
Yeah.
Not? Okay. Okay, so for now, I think the best plan is this one. Partly because of what I know of your classes, at least for most of you, that you will have physiology, right? And I think on Thursday that's true as well, but since we have 2 hours of PA and also the extra hour of meta, I think it's better to do it here. But again, let's just check for this one. Thursday 11 to 12. Anyone object? Okay, so I think both of these are options, and I'll ask Meta to finalize this with you on Thursday. And there's no extra class this week, but again, probably 4 times during the semester we'll have an extra class. This is what I expect. Okay, so what I would like to, yes?
There will be no class during the ISFM.
I know. Yeah, this is always true.
Okay, this is
And also there is a planned LSEC event. I don't remember exactly the date right now, which falls on a Tuesday, and we will miss 2 hours for that reason as well. Okay, so what I would like to do today is just mostly a background to tell you a little bit about the brain in general, although many of you already heard about these concepts. I think it's important in a course like this to start with a general background. And this will be followed up with a background on computational neuroscience, and then I'll go in a bit more detail to discuss what we're going to do in this course. So really the goal here is to make sure that everyone understands these basic concepts, that we have common terminology. Oh, I think that I'm supposed to use this because the classes are recorded. Okay, so let's start with neurons. Neurons are biological cells. They're quite, what is striking about them is, as you can see here in several examples, that they have very diverse morphologies. They're also very large cells. They include, like any other cell, an area which includes the nucleus of the cell, so this is called the soma of the cell, but they have these very long branches, and the branches are the way that these cells communicate with other cells. So, generally speaking, each cell in our brain has an axon, which is the output of the cell, and axons can be very long. They can go from the brain to our bodies. They can be in the order of 1 meter long. And the inputs to the cells are the dendrites. Often cells have 2 dendritic trees, and these can be very branched. And their length is much shorter, typically about 1 millimeter. The characteristic size is usually around 1 millimeter. And the reason, do you know what is the reason? Okay, so the reason is that the signal propagation, the electrical signal propagation in the dendrite is passive, and the signal decays as it propagates. And the decay length is approximately, ends up being about 1 millimeter. So a dendrite just cannot propagate the electrical signal faithfully over longer scales. Axons are different. In the axons, the signal propagation is active. The spike, we'll talk about spikes in a moment, are regenerated along the way all the time, and the axon is also myelinated, which helps increase the propagation length of the signal. Okay, anyway, as you can see, the dendritic trees are extremely diverse depending on types of neurons. In general, the brain is extremely diverse in terms of types of cells that it contains. It contains neurons. It contains also other types of cells called glia, but neurons are extremely diverse, and so probably in terms of just the number of types of cells, biological cells that exist in the brain is probably the organ that contains the largest diversity of types of cells. Okay, now from our point of view, neurons are the basic computational units of the brain. They receive inputs. The inputs are electrical. The inputs are mediated by connections between neurons, which are called synapses. And neurons produce spikes in response to inputs, and these spikes go to other neurons. Okay, I will not discuss here much the dynamics of how a neuron generates electrical spikes, but the important quantity which varies in a neuron, the most important quantity is electrical. It's the electrical potential difference between the inside of the soma of the cell and the outside, the membrane of the cell and the outside. And the dynamics of this quantity, the membrane potential, is quite complicated. It has to do with flow of ions through various types of ion channels and ion pumps. Okay, so what is a spike? Roughly speaking, when the membrane potential of a neuron crosses a certain threshold, roughly speaking again, a certain chain of events starts to happen. Opening of some ion channels, which very abruptly increases the potential of the cell, as you can see here. This is followed, this increase of the potential causes other events, opening of other ion channels that cause a reversal of this process, and the membrane potential goes back to some lower value and eventually goes back to baseline. So this is a very stereotypical event, meaning that the shape of this spike is determined by these fast processes that occur within the cell, and it's very, very similar each time the cell generates a spike. The width of the spike is about 1 millisecond. Okay? And these spikes, after they're generated, they propagate along the axons and they provide the inputs to other cells. Now, one of the central dogmas of neuroscience, perhaps like the central dogma of cellular biology, the DNA and the production of proteins, the central dogma of neuroscience is that spikes, the timing of spikes is what carries information. Okay, meaning that the shape of the spikes, which, as I told you, doesn't vary much, does not carry information. It does not carry information. It's just really the existence of the spike, the times at which they are emitted, that carry information. And we're going to believe this dogma in this course. And let's talk a little bit about how information is transmitted from one cell to the other. So I mentioned to you that the contacts between a dendrite and an axon are called synapses. And the one interesting aspect about how information is transmitted is that although most of the time information processing and transmission in neurons is electrical, on the synapse it's not electrical. When the spike reaches the dendrite, this causes vesicles, which are small membranes which engulf a certain chemical compound called a neurotransmitter. The spike causes these vesicles to be released. These find receptors on the axon side. Sorry, we went from the axon to the dendrite. The axon, the spike arrives at the axon. Vesicles are released, and these reach receptors on the dendrite. And the receptors, once the neurotransmitter reaches the receptor opens up ion channels, and therefore the signal is converted back, the flow of ions converts the signal back to an electrical signal. Okay, so here's just a very nice picture, electron microscopy picture in which you can actually see these vesicles being generated and released into the area between the axon and the dendrite. Okay, so we talked about this. And now, roughly speaking, we can divide these receptors into two types. There are receptors where opening of the receptor causes a decrease in the potential of the cell, okay? And there are receptors in which we will call a polarization of the cell, a hyperpolarization of the cell. Or we can have cases where the receptor causes flow of ions that increases the potential. If it increases the potential, it makes the cell more likely to spike. If it decreases the potential, it makes the cell less likely to spike. So synapses can be either exciting synapses or depressing synapses, and this depends on the neurotransmitter and the receptors. Now, one important law which we should mention is that cells, the presynaptic cell always forms one type of synapses to the postsynaptic cell. So a presynaptic cell is either excitatory or inhibitory. It cannot be excitatory to some cells and inhibitory to other cells, and I used the words always and cannot, and this is not true. Like any rule in biology, there are some exceptions, but overall this is true.
But you say the difference is the receptors, or?
It's both. It's a neurotransmitter, and the neurotransmitter activates certain types of receptors. Okay, so that's why it goes hand in hand. But it's really the. But one neuron releases one type of neurotransmitter. There are some exceptions to this rule. Okay. From our point of view, what this means is that neurons will be either excitatory or inhibitory.
But all neurons have both types of receptors.
Neurons, that's right. The neuron can have both, and most of them do, have both excitatory synapses and inhibitory synapses where they're receiving signals which are either excitatory or inhibitory. Okay, so now I just touched very, very briefly on biology, and of course the details are very complex and we're not going to go into them in the course, and what I want to present to you now is a very simple view of what a neuron does, viewed as a computational neuron, and this is more or less the level at which we will think about neurons in this course. A neuron receives input from other cells in the form of spikes. The neuron sums up these spikes in a linear fashion, maybe with some weights, and so the weights reflect the strength of the synaptic connection to the presynaptic neuron, and based on this set of, this chain of spikes that the neuron receives, and the sum of the chain of spikes, the neuron decides when to produce a spike itself. Okay, and usually, almost all of the time in this course, we will not even think about spikes explicitly, about the timing of spikes. We will imagine that the signal going into the neuron is a rate of spikes coming from a presynaptic neuron. So the neurons will think about the rate of spikes as a degrees of freedom, as the quantity that change over time. The neuron receives certain rates of spikes from other neurons. It sums these rates linearly, and this determines the rate at which the neuron itself is firing spikes. But in some cases, we will think about the spikes explicitly. Okay, so here's mathematically a simple rate model of neural dynamics. The details here are not very, very important right now. I just want to give you a feel for the level at which we're going to think about neurons. So what is determined by the neuron is its rate of firing. And this rate of firing is determined from a linear sum over synaptic inputs that the neuron receives from other neurons. So these are these Sj. Maybe there is some additional linear term which is just constant. So we will call this we can think about this term either as some bias to the activation of the cell, or as some external input which is coming from neurons which we're not explicitly modeling as part of the network that we're explicitly describing here. And all this linear sum determines the firing rate of the neuron through some nonlinear function. Why does this function have to be nonlinear? Here's some examples for this function, how this function could look like.
So there's no negative.
Exactly, because this is one obvious reason. Firing rates are always positive. So there has to be some kind of nonlinearity in the response of the neuron. In some cases, we will ignore this. We will imagine that the network is operating in a regime where everything is linear. If all the inputs, if these inputs are positive and sufficiently large, maybe this quantity never becomes negative, and we will imagine that this nonlinearity is simply linear. In other cases, this nonlinearity is important. And now, what can we say about the synaptic input coming from neuron J? So the synaptic input coming from neuron J is really determined from the spikes that neuron J is firing. But we should remember that when neuron J is firing, the current and at the soma of neuron I is not just a spike. When neuron J is firing, there is some current that exists over a certain characteristic time scale. And we can model this current in a simple way, just as an exponential function. So neuron J fires a spike here, and the current looks like this. This is a simple model for how the current will look like. So this is the current. This is time from the onset of the spike. And if we model this as something like e to the minus t over tau, then the characteristic time scale of the current is tau. And therefore, if the spikes, the synaptic activation is really a convolution of the spikes with these exponential kernels, and this differential equation really describes this dependence. So if you have spikes coming in at the times described by Rj, which are the spikes of the activity of the cell J, the synaptic activation is a smooth version of this input. So this is what this equation describes. Now, if we put these two equations together, we get the equation that you see here. Okay? And this is a time-dependent equation. This is a differential equation, which means that this equation describes dynamics. And so this equation can capture there are a lot of insights that you can obtain about what neural networks can do by just studying what sets of equations like this look like depending on the synaptic weights in them. So this is not really something that we're going to discuss much in this course. Maybe in Neural Networks 1 you discussed more dynamics. But in this course we will not discuss much dynamics of neural networks. So we often assume that our networks are at steady state. What does it mean that they're at steady state? It means that they may be receiving some constant inputs, bi, and then they settle after some time into a state where ds to dt is 0. And that, once this is true, you can see from this equation that Sj is simply equal to Rj, or Si is equal to Ri, and the firing rate of each neuron is given by this expression. So if we want to discuss static situations, a static input, and ask what is the relationship between the input and the output of the network, this was the input, this is the output. This set of equations is a simple description of what a set of neurons could do. Now, of course, this is a very simplified level of modeling a neuron. We could have modeled neurons much more detailed, perhaps modeling the dynamics of the ion channels in each neuron and the dynamics of the membrane potential. But in this course, we're not going to do this. Okay, now I want to shift attention to some basic concepts in computational neuroscience and ask what does a spike represent in the brain? And so much of our field has been dominated for many years by experiments in which one provides some stimulation to the brain, maybe a sensory stimulation, maybe some other forms of stimulation, and people measure activity of single cells by inserting electrodes into the brain and measuring the spike that they generate. And many interesting insights came from these types of experiments. And for example, Hubel and Wiesel showed visual stimuli to animals, to cats, and they noticed that if they record from neurons in the visual cortex, the first area that processes visual information in the cortex, for a while they had a hard time even understanding what makes a neuron fire. But after a while they noticed that if they show a bar with a certain orientation and they move this bar around, then cells start to fire. And a typical cell will be sensitive only if the bar is oriented in a particular way. So for maybe in this orientation it will fire many spikes, and if the bar has this orientation it will not spike at all. And then it's possible to plot what is the firing rate of the neuron in relation to the orientation of the bar. And this is what is called the tuning curve, and this is a typical tuning curve for a simple cell in the visual cortex. Here's another example. Monkeys were trained to move their arm in different directions, and a single neuron here was recorded in the motor cortex, and the observation was that the neuron fires more when the monkey moves the hand towards particular directions, fires less in other directions, and again we have a tuning curve. Now, does this mean that the neuron represented here, its role is to encode the orientation of the bar, or does this mean that the cell in the motor cortex, the role of the cell is to encode the orientation of the hand? What do you think? Not necessarily, right? It's part of what it's doing. It's in this particular experiment, this is what we showed the cell. This is what we asked the monkey to do. So of course the cell, I mean, the interesting thing, which is meaningful, is that this particular cell responds to this stimulus. Most of the cells will not respond to this stimulus if you show a bar in a particular location. Most of the cells in the brain will not respond in a very stereotyped manner to the orientation of the hand, but it's important to remember this doesn't mean that this is what the cell is doing. This is, in vision we have much more complicated stimuli than simple bars, and there's a long way to go from noticing that this type of stimulus strongly activates the cell and there is a strong dependence on the orientation, or that this cell's firing rate depends on the direction of moving the hands, to really understand how all the cells together in this brain area represent a stimulus, or how all the cells in this motor area are involved in a motor action. Nevertheless, these type of experiments are very important, and the concept of a tuning curve is a very important concept. Okay, here's just a simple way of going from one-dimensional stimuli. Here, why do I say that it's one-dimensional? Because the only variable that we're characterizing is the orientation of the bar. We might want to ask, what makes a particular cell in the retina, in our eyes, what makes a particular cell fire? So the retina is a very complicated neural network, and we will talk about it later in the course. It has 3 layers of cells. One of them is called photoreceptors. These are the cells that are sensitive to light. And the 3rd layer of cells, called ganglion cells, are the ones that send spikes to the brain. So all the brains know, everything that the brain knows about the visual world is really from the spikes that ganglion cells send to it through the optic nerve. Now, okay, so we can do an experiment where, well, I don't do this experiment, but other people do. They can take the retina of an animal out. The retina can still live for a few hours. You can put it on some solution which contains oxygen, and it lives. You can put it over an electrode array, record spikes from cells in the retina, and show the retina stimuli, and start to try to understand what makes a ganglion cell fire. So here's a simple model in which is often used to describe activity in the retina, where now the stimulus is not a one-dimensional stimulus. Now the stimulus is the whole two-dimensional picture. So it's a much higher dimensional quantity. It's the intensity of light at position x. Okay? And we can ask, what is the relationship between the intensity of light at position x and the firing rate of one particular ganglion cell? And it turns out that a model that looks like this works pretty well. So notice that we're integrating the intensity of light with some function. This goes through some nonlinear function, and this sets the firing rate. So this function that you see here is called a receptive field. And the first thing to note about it is that it's very localized. So a particular ganglion cell is sensitive only to the light intensity in a particular area of the visual scene. And if you look more closely at how this function looks like, you find typically that it's positive in some smaller region, okay? And it's negative in some wider region. So this is what is called the receptive field of an on-center cell. It's activated by more light intensity in the center of its receptive field, and it's inhibited by light surrounding this area. So mathematically, what does this mean that this cell is doing? What is it similar to? Yes, it's an integral, but notice that here it's with a positive sign, and here with a
negative sign,
So what is the cell sensitive to? Contrast, to a difference. It's similar to a derivative. The cell is really sensitive to the difference between the intensity of light that's here and the intensity of light in the surround. And one of the topics that we will discuss in this course quite extensively is why do receptive fields of retinal ganglion cells look like this? Why is it useful for the retina to encode the visual stimulus in this way, where it's not encoding simply the light intensity at every position, but it's encoding the difference between the light intensity at the center and the light intensity a little bit out of the center. Okay, so here are some of the concepts that we discussed so far: neurons, dendrites, axons, synapses, receptive fields, tuning curves. Okay. Now, what I want to do now is talk a little bit about computational neuroscience and the brain, and also provide some background on the brain on a bigger scale. So first of all, what is the field of computational neuroscience? So in Hebrew, חישוביות עצבית. So actually, this term is used in two different ways, and this is a little bit confusing. Sometimes when people say computational neuroscience, what they mean is simply using computational tools coming from math, physics, computer science to study the brain, okay? So any situation in which you use mathematical concepts in your work. But this is, in my opinion, this is not how I use this term. The way I use this term, and many others use it, is really the field where we're trying to understand the brain as a computational unit. We're trying to understand how the brain performs computation. Okay? Of course, in order to do that, we also need computational tools. We need concepts coming from computer science, engineering, math, physics. But our goal is to understand how does the brain perform computations, and how does the brain process information. So this is what this field is all about. And a single neuron can do a lot of things. It's a complicated object. Nevertheless, we believe that most of the computations done in the brain are the result of the collective activity of many neurons. And therefore, this field is often focused at the systems level, the level of systems containing many, many neurons. Okay, so let's talk a little bit about scales in the human brain. What I want to convey to you now is that the brain contains many, many different scales in several different ways. So we can start from the scale, from the length scale of the whole central nervous system, which includes the brain and the spinal cord. So that's around, in our bodies, it's around 1 meter. Let's go down to the brain and look at the cortex. The cerebral cortex is the part of the mammalian brain which we believe is involved in most higher forms of computation. And quite remarkably, if one starts to map this area into different areas, there are some common functions that you see in different parts of the brain. So, for example, the back of our brain is associated with vision. This part is associated with other senses. Here one finds areas that are involved in speech, reading, touch, and here motor cortex. So motor cortex is very much connected to the somatosensory cortex, which makes sense. Somatosensory is touch. And higher functions like decision making, speech are in the frontal part of the brain. How do we know that this is true? What do these titles come from? Okay, so for many years the main source of knowledge about these relationships was from lesions, either in people that had accidents, or in animals from lesions that are generated. One can see that lesions in particular areas cause loss of particular functions. And today, also to a very much large extent, fMRI, which allows to
relate activity in particular brain areas to actions, to stimuli, to various types of perturbations. Okay. So if we go back to length scales, we can say that in our brain, if we roughly call, this area, common areas that are associated with a common function system, then the length scale is maybe about 10 centimeters. But there's another length scale, which I will call the length scale of maps. I don't think that I have I think that I don't have a picture for that. But what do I mean by a map? For example, in the visual cortex, you could show stimuli in different positions, and you will find that there is a particular area, for example, V1, the primary visual cortex, where as you move the stimulus, you see responses in different areas of V1 arranged topographically. So, and there is a full area, there is an area of the brain which covers the whole visual scene. But there are other areas which also cover the whole visual scene. So each one of them is a map. Similarly, in the somatosensory cortex, you can find a relationship between position in the somatosensory cortex and the area in the body to which where cells respond to touch in that area. So this will be another map. So when I say map, it's the typical scale of a full map in the cortex. The length scale of the whole set of cells that map together, say the visual scene, is maybe 1 centimeter. Now we go down to the level of local networks. So there is an important and somewhat fuzzy concept in the cortex, which is called the cortical column. This concept comes from two types of observation. One is that local connectivity in the cortex is stronger over the scale of about 1 millimeter. First of all, actually, I need to mention something else about the cortex. I will not go into this in detail, but the cortex is a columnar structure. It is a layered structure. It's a structure that includes 6 layers, and you should think about the cortex as this 6 layers surface wrapped like a sheet of this 6-layer surface, wrinkled sheet. Okay, so now these are the 6 layers. Now, lateral connectivity is stronger in the cortex over a length scale of about 1 millimeter. So this sets a certain scale over which cells are more strongly coupled to each other than to cells outside. So this is an anatomical observation. It has to do also with the fact that, as we discussed earlier, dendrites, the characteristic size of the dendritic trees is about 1 millimeter. But there's another reason that people talk about cortical column, and that is that at least in some cases, for example, if you measure remember that we talked about the orientation selectivity of cells in the visual cortex of cats. If you look what is the orientation selectivity of a particular cell, you find that, and you map this for different cells, you find that cells within a region of about 1 millimeter share the same orientation selectivity. So cells within this area of a cortical column, at least in the cat visual cortex, encode something, all of them encode something similar. And therefore people think about the cortical column as perhaps some basic unit of computation in the cortex, but this is a debated question and it's a fuzzy concept, but nevertheless it's important. So the scale of this local network is 1 millimeter. The length here is maybe 3 millimeters. Okay, so now we go down in length scales to neurons. The size of the cell body of a neuron is maybe 10 to 100 microns. And I should mention also what do you see here. So this is a kind of a reconstruction of a cortical column, but we don't. The state of our field of neuroscience is that we don't yet have a full mapping of any single cortical column in the brain. There are very large projects going on in the world trying to achieve this, to map the full connectivity between and the structure of all the cells in the cortical column. What you see here is really an imagined cortical column. It's based on tracing the structure of many, many different cells, but then overlaying them together. We don't know, again, there's no case where we have the full connectivity in one cortical column, but this exists very soon. Okay, so going back to length scales. Neurons, 10 to 100 microns. Synapses, 1 micron. And of course, everything is composed of molecules, and the characteristic size of molecules in biological world is perhaps 1 nanometer. There's another light scale here, the size of an ion channel, which is composed of a few proteins, maybe 5 nanometers. So there's a very large range of scales going from 1 nanometer to 1 meter. And there is structure at every one of these scales. The scale that you should think about as the most relevant to our course and also to Neural Networks 1 is the scale of the local network. We really try to understand how a local network of cells achieves a certain particular computation. Okay, now let's talk about timescales, and then we'll take a break. So the timescale of action potentials I mentioned earlier is 1 millisecond. Membrane integration time. Remember that I mentioned once a spike is emitted, the synaptic current persists over a certain time scale. So that time scale, this is the synaptic integration time here. That time scale can vary depending on the type of synapse from 1 to 100 milliseconds. There's another time scale in the dynamics of the cells which I didn't mention explicitly when I wrote an example for the dynamics of neural dynamics, but it's possible to put it in, and this is the membrane integration time. What does that mean? If you provide if you perturb the membrane potential of a cell, you can ask how long will it take until the membrane potential goes back to its baseline value. This depends on the flow of ions through ion channels, and this is typically in the order of 10 milliseconds, a few milliseconds to 10 milliseconds. Okay, so 1 millisecond, 10 milliseconds, maybe some synapses generate currents that persist over 100 milliseconds. And now we go from dynamics to plasticity. Plasticity is the change in the strength of connection between neurons. And we know that there are different types of plasticity. There are types of plasticity which are short-term, they are transient. So when a cell fires many spikes, when a synapse is activated many times, it might be tired. It might fire less spikes over some time afterwards. Or the opposite could be. Some synapses are, in some cases, when a synapse fires spikes, the next one will cause a larger current. So the characteristic timescales of short-term plasticity range between this range of about 100 milliseconds to a few seconds. Okay? These are processes, so the synapse, the current state of the synapse, how strong is the synaptic current that it's going to generate the next time it's activated, depends on the history over these timescales. There's another type of plasticity, which is long-term change in the strength of synapses. And even though there are some examples where we know that the strength of long-term changes in the strength of a synapse can arise from very brief events. For example, a single activation of a cell in the hippocampus has been shown recently to change its receptive field. Most of the time we think of long-term synaptic plasticity as a very slow process that requires many, many spikes and over a time scale of hours. Okay, so 10 to the 4 seconds.
If it's one event, it still takes time for the synapse.
Yes, and in the example that I mentioned, I don't know how much time it takes, but it's quite fast. But generally, we think about long-term synaptic plasticity as a process that requires hours. But the outcome of this process is memory that can last a lifetime, right? So we know that there are long-term structural changes that are dependent on our experience, which last for 50 years. Okay, so again, you see here a very wide range of time scales. And some of them are kind of mixed with each other. So, short-term synaptic plasticity is not so far in its time scales from simply the synaptic integration times and the long-term synaptic plasticity. Okay, maybe we should take a break now, so we'll continue afterwards. I'll continue at 1.
Uno sta nelle posto. Vecchia.
כן, האמת שזה טוב שזה חיבור, רגע. a very bad memory for names, but I would like to try to remember your names. So one thing that I'm going to do is I'm going to take a picture of you now. When you ask questions, please mention your name and I will forget it. But next time I will also write it down on the picture. And hopefully by the end of the course,
Just so you don't get embarrassed. What's your name again?
Yoram. Okay. So we were talking about length scales and time scales, and now I want to talk a little bit about number of neurons. So number of neurons in different nervous systems vary by a lot in different animals. C. elegans. So you probably know that in biology there are just actually a handful of organisms that biologists study most of the time. C. elegans is one of the most important model organisms for a multicellular animal, and it has 302 neurons. One of the remarkable things about C. elegans is that its whole body plan is completely genetically encoded. So every two individuals have the same number of cells, the same arrangement of the cells, and this is true also for the nervous system. So each cell in C. elegans has a name. And so there are 302 neurons, and with these neurons, the C. elegans moves around, senses its environment, learns a little bit, and so on. Number of synapses is about 5,000. Drosophila, another very important animal studied in biology. Fruit fly has, now we go up by 2 orders of magnitude, has 10,000 neurons and about 10 million neurons. So you see that the ratio between the number of synapses and neurons is here about 100. Here it's a little bit more than 10. Now we go to the mouse, which is the model animal for mammals. Go by a factor of 1,000 in the number of neurons and a factor of 10,000 in the number of synapses. So now the ratio is about 1,000 between the number of neurons and the number of synapses. Go further to humans, another factor of 1,000 and a factor of more than 1,000 in the number of synapses. So in the human brain, the ratio between the number of synapses and the number of neurons is closer to 10,000 than 1,000. Okay. Now if we look specifically at the cortex, which as I mentioned is involved in most higher functions in our brain. Cortex contains about a fifth of the neurons in our brain, so it's about 2 to 10 to the power of 10. And we zoom down to the level of cortical column, which as I mentioned is about 1 millimeter to 1 millimeter on the surface, and the depth is about 3 millimeters. Cortical column in the human brain contains about 10,000 neurons, and 10 to the 7 to 10 to the 8 synapses. So this means that each neuron in the cortical column, each neuron in the cortex, on average, receives about inputs from about 10,000 other cells. Okay. So this leads me, this observation that we have so many cells in the brain and they're so interconnected, leads me to discuss some of the challenges of computational neuroscience. The dynamics of the brain and the function of the brain is achieved through the collective dynamics of many units, many neurons. We need to understand the collective dynamics of a very complicated system which contains many degrees of freedom. As we discussed, there's a vast range of time scales and of spatial scales. And one of the difficulties in biology in general and in the study of the brain is that there's complex structure on all these scales. Most of it is unknown, and it's often a very difficult question to even formulate to ourselves our approach. What details are important? When we want to understand how we perform a certain visual task, do we need to model the activity of neurons down to the level of ion channels? Can we ignore that and model the neurons at a more coarse level? Are there some functions where we can model at an even coarser level? Okay? So what level of description is important? That's a very difficult question often. What makes
the brain different from special compared to other biological systems is its function. What is the function of the brain?
So it's supposed to be.
Also complex, but I will argue that it's computational information processing. And evolution was free to tweak the properties of the brain on all the scales that we discussed in order to achieve function. So this is really what makes biology different from other systems. So I'm a physicist in my training. Physicists know a lot about how to model systems that contain many, many degrees of freedom. This is what the field in physics which is called statistical physics, where we study a solid or a gas which is composed of 10 to the 23 molecules, and we understand the properties of them. But what makes biology very different from most physical systems is the fact that there is complex structure on so many scales. And the structure has been tweaked by evolution to achieve a certain function. And often there are many different mechanisms that contribute to a certain function, acting on different scales. Okay, so what are the computations that the brain does? So here this is just a list of some examples. Sensory perception, we talk a lot about sensory perception in this course. Object recognition is not something that we'll discuss in detail, but discrimination, the early stages of sensory perception, decision making, the generation of motor output, which involves setting up an appropriate, complicated, time-dependent set of outputs to eventually to the muscle, memory, learning. These, of course, are related to each other, and we can go up to higher functions, learning, language, creativity, and so on. So the brain does all these things. One thing which is common to every aspect of what the brain does is that information plays a central role. All these computations involve representation of information, extracting of some information, maybe discarding of some information which is not important for the function, relaying of information between different brain areas, and storage of information. And therefore information plays a very central role in computational neuroscience. And the term learning, which I used, is used in various ways in neuroscience, so it's important to clarify a little bit that the term is used mostly in two different ways. One is from a mechanistic point of view. Often when we say learning in neuroscience, really mean any process that involves long-term changes in the connectivity of the brain, the strength of synapses and the connectivity. And so it doesn't necessarily, from this point of view, so often we say learning without necessarily identifying exactly what is being learned. And another term which is more correct, perhaps, for this aspect of learning is plasticity. And then there's a functional point of view. Now, from the functional point of view, when we are going to model learning in this course, we're going to make one very important distinction, which is a little bit artificial, but it's important, and this is the distinction between what we will call supervised learning and unsupervised learning. So supervised learning is a situation where you have a set of, you need to learn a certain association between some examples and some output, identify what are cats or what are dogs, but you're being told. Whenever you're shown an example, you're being told, this is a cat, this is a dog, and you want to learn this relationship. Unsupervised learning is a situation in which, imagine again that I would show you many, many different pictures of cats and dogs before you ever saw cats or dogs. I will just show you these sets of pictures, and your brain, by looking at these pictures, will reach the conclusion that there is a categorization. There's two different categories. One is cat and one is dog, without anyone telling you which is which, okay, in each example. And there are some forms of learning that we will formalize, that can be formalized, which are a little bit in between these two extremes. Okay, now I want to mention a little bit some of the assumptions that we were going to make in the approach that we're going to take in this course to modeling computation in the brain. The first one is that we're going to focus on models in which neurons are described in a very simple manner. We're not going to model explicitly the ion channels of neurons and not the spike generation. We're going to model, as I already showed you before, we're going to model neurons in a very simple way. So our assumption is that the interesting properties of dynamics and learning in the brain arise from the network, from the way the neurons are connected to each other, and not from the intrinsic properties of individual neurons. And the other assumption is that learning is achieved through changes in synaptic connectivity. Okay, I've already showed this equation. This is just a typical example for how we're going to model neural dynamics in this course. And even this is more complicated than what we'll do most of the time, because often, as I mentioned earlier, we're going to look at steady state. And in some cases, we're going to ignore this nonlinearity. We're going to imagine that the neuron is really just doing a linear computation over its input. And this is a very rough representation of the kind of assumptions that we're going to make about plasticity in the brain. So the idea is that the change in the connectivity between neuron J and J, is some function of several things, but these things are mostly local. So what do I mean by that? The change in a synapse mechanistically can depend on the firing rate, on the activity, let's say the activity of the presynaptic and postsynaptic neurons. When I say activity, there are different levels of modeling. We might imagine that all that matters is the firing rate of the pre and postsynaptic neurons. We might imagine that what is important is the precise timing of their spike. So I'm not committing here in this equation to a particular approach, but I just want to give you the flavor. The change in the synapse depends on the activity of the pre and postsynaptic neurons. It might depend on the state of the synapse itself. All this is local. In some forms of where we approach plasticity mechanistically, we also imagine that there is some kind of reward signal. This reward signal influences whether the synapse is going to strengthen or decrease. And do you know biophysically what can be the manifestation of this reward signal in the brain? Okay, neuromodulators. Okay, so neuromodulators influence, they do several things, but among them they influence plasticity. So a splash of neuromodulator might make a synapse strengthen in a situation in which it would not strengthen in the absence of a neuromodulator. And maybe there are some other local variables, and here with the prefactor here, lambda, which I call the learning rate, is simply just a prefactor in terms of how fast learning is happening. So this is a kind of mathematical description that we will use when we want to discuss plasticity mechanistically at the level of individual synapses. Okay, so I think now I can go through the syllabus, and then we'll start a little bit to talk about some materials. So before I present the syllabus, I want to mention some of the broad questions that we will address in this course. So a large part of this course will deal with neural coding and decoding. We will ask, what are the limitations? How does the way that neurons represent information influence the ability to decode this information from the joint neural activity? Joint neural activity meaning that we will not be interested necessarily in just being able to read out something from the activity of one neuron, but we'll ask, how can you read out some quantity from the joint activity of many neurons? And how do we characterize the way neurons represent information? And very importantly, a large chunk of the course will ask whether there are some principles that determine how the brain encodes information. Remember that I showed you this example of the receptive field of the ganglion cell in the retina, the center surround receptive field. These are some principles behind the fact that ganglion cells represent information in this particular way. And the answer that we will discuss a certain hypothesis, which is a hypothesis called the efficient coding hypothesis, hypothesizing that the way that neurons represent information is set up to efficiently represent this information. This will become more clear later in the course. But since I'm talking about efficiency, this is related to some optimization principles. Argue that there are some optimization principles related to efficiency of information representation that can explain a lot of the phenomenology of how neurons represent various quantities. And after we will identify some optimization principles, we'll not talk about this a lot, but we will discuss whether local plasticity rules acting at the level of individual synapses can give rise to a network that efficiently represents information. So this is a form of unsupervised learning. We will also discuss learning in other ways. Again, part of it is how do local plasticity rules shape the structure of a neural network, but part of this will be asking what kind of computations can be done by a neural network which has a fairly simple structure, neurons summing up their inputs, and how can these computations be learned. And when we will discuss learning, which is a form of plasticity, some of it will be at a more abstract level without asking, we will ask if we want to update the weight of the neural network such that it performs a certain computation, how can we do it from a more algorithmic point of view? And then we might talk a little bit about how local plasticity rules might implement this computation. So this is more dealing with supervised learning, this is more dealing with unsupervised learning. And I think this I already had before, so it's just based in our own place. And so some of the main themes in this course are, as I mentioned, neural coding and decoding, learning, and one thing to note is that we will have very little discussion on dynamics of neural networks in this course. There was much more dynamics in neural networks 1. I mentioned this also in the context that most of the time we will think about neural activity as some static output in relation to some input. Very little dynamics. But there's discussion on dynamics at the level of the synaptic weight, because this is what is going on in learning. We're thinking about neural networks as computational devices, as some collection of cells that have an input, receive an input, generate some output. Another important theme is going to be noise. Noise is going to play a very central role, because if we ask, for example, how faithfully can you read out some quantity from the activity of neurons, the fact that there is a limitation to how faithfully you can read it out has to do with the fact that the activity of each neuron is noisy. If it was not noisy, we would be able to perhaps read out this quantity to any degree of accuracy that we want, but the activity is noisy. Another important theme is local plasticity rules, as we mentioned, and the fact that computation and learning are emergent phenomena that arising from the collective dynamics of all the synapses or the neurons in the network. Okay, here's the more detailed syllabus. So this year I switched a little bit the order in which I'm teaching things compared to the past 2 years. I'm going to start with supervised learning. Very, very basic concepts on learning, which are quite old. It's really just an introduction to how you can learn various functions in simple neural networks. Everything with a question mark is something that I'm not sure that I'm going to do, partly because our semester has been shortened a little bit. After we do this introduction to supervised learning, I will spend time on neural decoding. So we will ask, I mentioned this before, but I'll say this again. Imagine that you observe the activity of many different cells, and we have some assumption about how these cells represent a certain quantity, the tuning curves of the cells if you want. We will want to ask, suppose that you observe the size of the cell, how well can you read out the quantity that they're representing? So we will discuss discrimination tasks, where you need to, say, distinguish between a bar disorientation and a bar disorientation. We will also discuss estimation tasks, where your goal is to read out the angle of the bar. And so there are some interesting relationships to concepts in statistics, which we will discuss. Population coding, because most of the time the readout is not from one neuron, it's from many neurons, and some other forms as coding. We'll discuss a little bit whether correlations in the spikes of different neurons are affect the ability to decode, and some particular forms of decoding algorithms. So here we're focusing on decoding. The next part of the course, which is a very significant part of the course, two parts are strongly linked to each other, is just shifting the attention from decoding to coding. We want to ask, why do neurons encode information in a particular way? And we'll start from the efficient information hypothesis, which was posed by Horace Barlow. We will discuss a little bit information theory, which is a very important framework for discussing these questions. So here's the part which will be kind of a bit mathematical and formal. And then we will discuss how this hypothesis relates to coding in the brain. A lot of emphasis on the visual system, trying to understand central surround receptive fields in the retina and the receptive fields of neurons in the early visual cortex. So much of the topics that we discuss here from the point of view of how does the brain encode information have to do also with other concepts which are often used in neuroscience, which is dimensionality reduction, because the framework that we will discuss here often will be a situation where we have a very high dimensional signal going into some neural network, like the visual inputs, and the representation of the signal by reduced number of neurons. This is a form of dimensionality reduction. So our discussion of this topic, of these questions, will lead us to some important algorithms for dimensionality reduction, which are used also in many contexts outside of neuroscience, like PCA, principal component analysis, or dependent component analysis. And the question of how a neural network arranges in order to perform these computations is a question of unsupervised learning. And then I'm not sure I'll have time to do this, but I might talk a little bit about some more recent work that this is actually work that we've been doing in my group, which is related to spike time independent plasticity. Plasticity in neural networks involves the precise spiking of pre and postsynaptic neurons, and how this can shape the structure of the neural network. Recommended books, so there's a list that I will put in on the Moodle, but some of the more relevant books that you can use. So this is probably the most current comprehensive book on theoretical neuroscience, which just presents this field in a contemporary way. It's not a very deep book, doesn't go deeply into most questions. It's not a very difficult book to read in terms of the kind of math that it uses, but it's a good overview of the field, and it has chapters that are relevant to things that we're studying in this course. This is a more mathematical book, and also an older book, which is useful. This one is just a nice book to read. It's less relevant to particular topics that we will discuss in this course. And it's by now also not a very new book. It's very much focusing on encoding in single neurons, but I think that it's a nice book to read from which you can get a nice perspective on questions of coding. And this is a standard textbook on information theory, so when we reach this topic, this book can be helpful. Okay, so I think this ends my keynote presentation. Any questions? As far as I remember, what we did in the past 2 years is that it's open material in some way. I think I'm trying to remember if it's we don't allow you to bring notes or only allow you to bring notes. It's what I don't allow you. One of these two. So I just don't remember what we did. I'll check and I will let you know.
Do you put the presentations in the Moodle as well?
So I will put this presentation on Moodle, but all the rest of the course will be on the whiteboard.
Ah.
Okay, so this is really a very unusual lesson today. But
assignment?
Next.
Daniel.
Daniel and Oren.
Assignments. How many assignments are there? When do we get the first assignments?
There was in the beginning 6, 7.
And
There was at the beginning 6 or 7.
yeah, so typically it will be once every 2 weeks. And as far as the, how it's 15% of the grade, but I don't remember from whether we take all of them or all the best except one. Neta will be able to tell you in more detail after I discuss it with her.
Okay. In case one misses one or few times, one has just to catch up on his own. Lectures will be recorded on YouTube.
So lectures in general, everything that's going on in this room is recorded, and they're supposed to be also posted. Lectures should be posted, but I'm not sure actually this year who is in charge of this. So this is something to check. And it's a good thing that you're reminding me to make sure that it's actually being done.
Yeah. Also, so in future, it's probably better to speak to the microphone because of the.
Oh, microphone.
I mean, now it's organized.
Yeah, today it's less critical, but
Yeah.
I agree. So please, if you notice that I'm not using the microphone, please remind me. Because here in this class, it's not necessary for, just for the
recording.
It's just for the recording. Any other comments, questions? Okay, so the real part of the course is now starting because we're going to get rid of the screen.
Date of the exam?
It's probably already set, and therefore it's probably on the shnaton, the.
Probably. It's not up here.
Okay. Okay, so I need to erase the whiteboard. So I'm going to start now our discussion on supervised learning. And what I will do today is really just set up the framework, just some terminology and explain how we're going to approach the question of supervised learning. Okay, so the setup that we're going to have in mind is that we have some neural network, and this neural network receives some inputs. I'm going to denote the inputs by a vector x, so you should think of this as the input to each one of the input neurons of the network. And the neural network generates some output. And I'm going to denote the output by y, which could be a vector. Okay, it could be the activity of several units in the network. And what is this output going to depend on? It's going to depend on the inputs. And it's going to depend on the synaptic weights within the network. So remember, our framework is that we're thinking of learning as a result of changes in synaptic connectivity, the strength of connections between neurons. So we're going to denote that by a matrix W. In the slides, I denoted this by J, but here I'm going to, most of the time, I'm going to use W. So W represents the synaptic waves. And our goal is for the network to learn to represent a certain function, so, or approximate a certain function. This function itself might be multidimensional, but most of the time in the example that we will discuss, it will be just a one-dimensional function, so therefore also the output, we'll think of the output as just the activity of one output unit in the network. So most of the time, Y will not be a vector. Okay? And so, again, the goal is to somehow learn to set up the synaptic weights such that the relationship between Y and X will approximate well this function f of X. Okay? Now, whether or not this is possible at all depends on many things. It depends on the function, and it also depends on the architecture of the neural network, because the only thing that I allowed here to vary are the synaptic weights. But the neural network will have a certain architecture, and whether or not, and there are certain aspects of, there are assumptions on what is the relationship between the output and the input of each neuron, which will play a role. So whether a certain network with a particular architecture can approximate well a certain function depends, among other things, on the architecture. Now, what do we mean by supervised learning? What we imagine is that we're providing a bunch of examples, a set of examples. So we have examples. We're going to label each example by a label mu. And so from here on, I'm imagining that the function is one-dimensional. So x, the input is a vector, the output is y mu, and in the examples, we're being told explicitly what is the value of the function for this particular x mu. Okay? So each example is an input and a desired output. And our goal is to adjust the weights such that y of x mu w is approximately equal to f to y mu for all the mu's. What do I mean by approximately equal to? Why am I saying approximately equal to and not equal to? Because possibly particular network architecture cannot exactly, even if we find the best possible weights, maybe it cannot exactly reproduce this function, but we want it to approximately reproduce it, and approximately means that we have to have some measure for how well we're doing. So the precise meaning of this statement, that it's approximately calculating the function, depends on our measure of what is good, what is close, and what is not close. So this will depend on how we decide to measure this. And there are various variations on this theme. For example, what type of variable is the output y? In some cases, we will imagine that the output is a graded output. Can take any value within a continuous range. In other cases that we'll discuss soon, we will be interested in a binary output. The only output of the network will be either the y will be either a 0 or 1. This corresponds to a categorization problem. For example, we might imagine that the network receives a set of inputs that represent an image, and it needs to decide, 1, if the image contains a cat, and 0 if the image does not contain a cat. Okay? Things depend on the measure of error. And now another thing to note is that there are different questions that we can ask. We can ask, what is the best performance that can be achieved for any choice of W, but we can also ask, what are some algorithms that we can use to adjust W to achieve the goal, and how well they're going to do. And this, as I mentioned.
How do you separate the two Y's, like in the last line?
Here?
Yeah, like.
There's one Y here.
And
Oh, yeah. Okay, so when I write a Y without a mu, I just mean the output of, so there are two functions here. Okay, one of them is the function that we want to approximate. This is f of x. The other one is the output that a network is generating for a particular x. This is also a function of x, right? Also a function of w. So when I write a y without a mu, I mean this function, the relationship between the input of the network and what it spits out. When I write a y mu, I mean the value that was given in this example. So this is not a notation which is maybe a little bit confusing, but it's common to use it, and so I'll follow this notation. Okay, it's a good question. Okay, so we really have just 10 minutes, so I think I will start to discuss the first example that we will deal with, but most of this will be finished next week. Can you see? Can everyone see this? Yes? Because this is could be obstructing. So in general, if I write in this area, where does it start to be a problem for those of you who are in this part? Here it's okay? Here it's okay? Here it's a problem already? But if I go lower, it's a problem. Okay, I will try. But if I do it, if I write it, let me know, because I sometimes might forget about it. Okay, so the topic, the example that I want to start with is perhaps the most simple manifestation of this problem, and this is a linear problem. So what do I mean by this? So let me write the title: linear networks, and sometimes this is called the linear perceptron. So imagine that the architecture of a network is the following. We have a set of input units, x1 to xn, and we have one output unit, y, and y is connected through synapses, if you want, to all the input neurons. So let's denote the strength of the synapses, and we have just one output unit. Our synapses form a vector. We can label them by W1 up to Wn. Okay, so these are the strengths of the synapses. And the input-output relation that I'm going to assume is simply a linear computation. So y is going to be a sum over I going from 1 to n, wi xi. That's all. So the input is graded, the inputs are continuous variables, the output is a continuous variable, and it's simply a linear sum over the inputs. And I could write this as w, in vector notation, as w transpose times the vector x. So this is what the network can compute. It can only compute, our network can only compute linear relationships. And now we want to learn a relationship which is not necessarily linear. Y is some function of x. This is the relationship that we want to approximate. Scalar function. And we're learning from examples. X mu, y mu. Mu goes from 1 to p. So there we have p examples where y mu is f of x mu. And now another thing that is important to formulate is the questions of where are the examples coming from. So what we're going to imagine is that there's some probability distribution p of x. So remember, x belongs to R to the n. X is a vector, n-dimensional vector. And we're going to imagine that when this learning is going on, someone is randomly choosing examples from some distribution over x. And I'm not going to specify it now explicitly, but this is going to be somewhat important, right? Because obviously, all we can hope to do is to learn the relationship in those areas of Rn which are sampled by this probability distribution, for example. Okay, so before we end the class today, what I want to formulate is to formulate this goal a little bit more clearly. As we mentioned, in general, we probably cannot exactly learn this relationship. If this relationship is nonlinear, a linear network will not be able to realize it. And so all we can hope is to realize it approximately, and we need a certain measure for how well we're doing. So there are various ways to quantify this. And the one that we're going to assume explicitly is a mean square error. So we're going to imagine that we quantify the error as follows. So this will be a measure for a particular example, a measure of error for a particular example. Okay, so this is quadratic measure of the difference between the desired output and what our network is actually doing. It's going to be 0 if our network is doing precisely what we want. The half is just for convenience. And so we can ask what is this error for each one of the examples. So this will be epsilon mu, which depends only on W and on mu, and this is half y of x mu W minus y mu squared. And we can also quantify the total error over all the examples by summing up the errors over over each one. So we will call this error, I'll explain the TR that I'm writing here in a moment. We will define a global error over all the examples, which is going to be the average error per example. So it's 1 over p times the sum of mu going from 1 to p of epsilon mu of W. And this is called, I'm going to call this the training error. This is a quantification of how well our network is performing over the training data set. Okay? So as you can imagine, a possible goal for our learning would be to minimize this training error. Okay? So we'll continue from here on Thursday.
Okay, the question was why I'm labeling it only by mu, because mu is already related to the set of examples. There's an x mu, there's a value, so mu also tells me what is x mu.
That total error is the training error?
So the training error, if you wish, it's the average error, total error over the training example. It's the sum over all the training examples, the error for each one of them, and I divide by 3 because I want
training errors for each example there is a different training error?
No, because the training error is this sum. Okay?
So it's the same
I
as
just
the
use
total error?
yeah.
Oh, okay.
We'll discuss next time how the examples have to be made, because the examples of the training set are bad.
Yeah,
Then we can minimize the training error, but if we have different cases and example, then
yeah. So actually, I will not discuss so much in terms how to choose the examples, but I will discuss some formal questions. How many examples can you deal with and set the training error to 0, and what will be your error on other examples?
I mean, for example, we have a threshold linear function and we want to approximate it, and then we only take the 0 part. Very easy to...
Exactly. So there are many, the theory of learning is a deep mathematical topic, and I'm not going to go into detail, so I'm just going to discuss some basic algorithms.
Does the person learn anything from the previous project last year?
You can. Does what?
Does it rely on anything?
Not so much. It's not a little bit conceptually, but
אוקיי. עשיתי אותה. לא, האמת שאני שואל את החברים משנה, כאילו, אתה יודע גם עד כמה זה...
אני בעיקר ממליץ, אנשים שבאים מרקע מתמטי, אני חושב שלא צריך להיות קושי לקחת.
השאלה עד כמה זה שונה מדברים במדעי המחשב נניח, של לרנינג.
אז החלק הזה של הקורס, 4 שבועות הראשונים, מכוסה גם בקורסים שלומדים במדעי המחשב, ויותר בצורה יותר מזה.
פה או שם?
שם.
שם, אוקיי.
שם, לפי דעתי כן. אבל בהמשך הקורס...
Machine learning?
Machine learning, האמת שיכול להיות שאפילו פה, חלק מהנושאים ביגוד מידע במידע, תלוי קצת על השם הקורס. המשך הקורס, הייתי אומר שלא. כל מה שקורה בהמשך הקורס, אני חושב שלא נדבר בכלל.
יש לפעמים חפיפה, כי הדיון באיך לשלוט מאוד בנקודת אינפורמציה יוביל אותנו